Hello everyone and welcome to the new session of the lecture series, and today's topic is respiration in plants. So today what we will do, we will revise the entire chapter and all the topics covered in this lecture are totally NCERT based. Alright. So let's start with the respiration. Okay. So respiration is what? Respiration is a metabolic process that involves breaking of the carbon-carbon bond, okay, of the complex organic molecules, or you can say compounds, alright, which are known as the respiratory substrate. So what is happening in respiration? It is a catabolic pathway, okay, which means the complex organic compounds, okay, are broken down into simpler organic compounds, okay. Here we have complex organic compound broken down into simple organic compounds, and in the process what happens? Energy is released in the form of ATP. Alright. So this is known as the respiration. It is a metabolic pathway. Okay, you have to remember these terms first: metabolic pathway, then breakage of carbon-carbon bonds. Okay. Then you have to remember complex organic materials are converted into simple organic materials. Okay. And there is a release of energy in the form of ATP. Now, respiratory substrate. Okay. What are respiratory substrates? These are the compounds that are oxidized during the respiration. And usually glucose is used as a respiratory substrate, but depending on the requirement of the organism, other respiratory substrates such as fat, proteins and organic acid can also be used as a respiratory substrate. Now let's discuss about the features of the respiration. First, it is an oxidation process, means oxygen is generally utilized in the respiration process, okay, and generally during the aerobic respiration. Okay, in anaerobic, oxygen is not required. Okay, second feature is catabolic process. Okay, means there is a breakdown of the complex organic compound into simple organic compounds. Okay, and third one is it is an exothermic reaction, means energy is released during respiration. Alright. Now let's discuss about the structure of the mitochondria. So mitochondria is a double membrane structure, means it consists of outer membrane and an inner membrane. Okay. Then we have the matrix. Okay. Matrix is what? It is the fluid of the mitochondria. Alright. And which consists of all the enzymes responsible for the biochemical pathway, and is also consist of the coenzymes, okay, such as NAD, NADH, okay, FAD, okay. Now this one is the crista. What is crista or cristae? These are the infoldings of the plasma membrane or the membrane. They are the infoldings of the membrane, and what does they do? They increase the surface area for the biochemical reaction. They increase the surface area. Alright. Now let's first talk about the aerobic respiration. So aerobic respiration, okay, consists of three steps, you can say. The first one is, or you can say three pathway. First one is glycolysis. So glycolysis is what? It is conversion of glucose into pyruvic acid. Okay, and this takes place in the cytosol or the cytoplasm of the cell. The next pathway is the Krebs cycle. Okay, and this takes place in the matrix of the mitochondria in eukaryotes. Alright. So what happens? This Krebs cycle includes one step, okay, or you can say there is one step before Krebs cycle, which is known as oxidative decarboxylation of pyruvic acid. also known as oxidation of the pyruvic acid. Alright, so we will look all these pathway in detail, okay, in coming slides. The next is the oxidative phosphorylation, okay, or you can also say electron transport system. So oxidative phosphorylation, okay, involves formation of ATP, alright. And where does this electron transport system or oxidative phosphorylation takes place? This takes place in the inner mitochondrial membrane. So if we talk about aerobic respiration, we have to mention all these pathways and mechanism. Now first let's talk about the glycolysis in detail. So glycolysis is also known as EMP pathway. Okay. Embden, Meyerhoff and Parnas pathway. Okay. So these were the individuals which gave the glycolysis pathway. So this is the glycolysis pathway where a glucose molecule, which is a 6-carbon compound, is finally converted into pyruvic acid, which is a 3-carbon compound, and 2 molecules of pyruvate or pyruvic acid are produced. Here you can see glycolysis is a 10-step process, okay, or 10-step pathway, and it consists of 10 enzymes, okay, which are present in the cytoplasm. Alright, so till stage 5, okay, or till step 5, okay, or you can say till the formation of glyceraldehyde 3-phosphate, till here it is known as the preparatory phase. Okay, means preparatory phase. Okay, and during this phase, what happens? ATP is consumed here. You can see these are the phosphorylation step where ATP is consumed. Okay, and later on after the preparatory phase, what happens? ATP is synthesized. Okay, so this is the breakdown of the glycolysis pathway. So what happens? Glucose is converted into glucose 6-phosphate. Alright, ATP is consumed, and this step is known as dephosphorylation step. Okay. Again, what happens? Glucose 6-phosphate is further converted into glucose 6-phosphate. Enzyme involved is phosphoglucomutase, or you can say phosphoglucoisomerase, and this step is an isomerization step. Okay, so what is happening? Glucose 6-phosphate isomerizes into fructose 6-phosphate. Okay, the next step is again dephosphorylation. This is second dephosphorylation, and fructose 1,6-bisphosphate is formed. This is second dephosphorylation. Now what happens? Fructose 6-phosphate, okay, is broken down into dihydroxyacetone phosphate, okay, which is a triose phosphate, and glyceraldehyde 3-phosphate. Okay, these are triose phosphate, means they contain 3 carbon atoms. Okay, that's why triose, and a phosphate group. Okay, this reaction is catalyzed by the enzyme aldolase, which is a cleavage enzyme. That's why this stage is known as the cleavage step. Okay, and aldolase what do? It is a kind of a lyase enzyme. And what is the role of lyase enzyme? It catalyzes the cleavage of, okay, the organic compounds. Okay, so what is happened now? This entire is known as the preparatory phase. Alright, now let's continue with rest of the, okay, steps. Then we have the 6th step. Okay, this step is known as the oxidation step. Here what is happening? Glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate. Enzyme involved is glyceraldehyde 3-phosphate dehydrogenase. So this enzyme is an oxidoreductase enzyme. Oxidoreductase enzyme. What it does, this enzyme, this reduces one compound as well as at the same time oxidizes other compound. So there is oxidation reduction reaction going on here. And what is happening? NAD is converted into NADH. Okay. And what else is happening? Inorganic phosphate is being added to the glycerate phosphate, leading to the formation of 1,3-bis phosphoglycerate. Now next step, what happens is known as the phosphorylation step. Okay, this is known as the phosphorylation step. And what is happening here? Here substrate level phosphorylation is happening. Okay. Substrate level phosphorylation. What is substrate level phosphorylation? It is the formation of ATP, okay, from ADP, okay, or you can say formation of GDP also from GDP, okay, by the transfer of the phosphate group from high energy compound to the ATP or the GDP. Alright, this is known as substrate level phosphorylation. So now what we have, 3-phosphoglycerate is produced, which is, okay, converted into 2-phosphoglycerate. So here what happened? 3-phosphoglycerate is isomerized to 2-phosphoglycerate, and here what happens? There is a transfer of a phosphate group. Okay, you can also say this is an kind of isomerization step. Alright, and what is the role of mutase? Mutase, they cause molecular rearrangement. They cause molecular rearrangement and especially of the phosphate groups. Alright. Now coming to the next step, 2-phosphoglycerate is converted into phosphoenol pyruvate and a water molecule is released. Enzyme involved is a enolase enzyme, okay, and this enzyme, okay, what it does, it removes hydroxyl group, okay, that's why water is released here, okay, and this step is known as the dehydration step. Okay, and finally what happens? Again substrate level phosphorylation takes place. Okay, this one, 10th step, phosphorylation is taking place here, leading to the formation of 3-carbon compound pyruvic acid. Okay, and 2 molecules are synthesized. So here you can see what is happening. 2 NADH are being produced here. Okay, so what is the net gain? 2 NADH plus what is happening? ATP is being synthesized. Okay, 4 molecules of ATP are synthesized. Okay, 4 ATP are synthesized, but what is happening? 2 ATP were used or consumed during the preparatory phase. So what will be the net gain or net output of ATP? 2 ATP. Alright, so net gain of glycolysis is 2 NADH and 2 ATP. Okay, now pyruvic acid which is formed at the end of glycolysis can either enter the aerobic pathway or it can enter the anaerobic pathway. If it enters the anaerobic pathway, then okay, what happens? There is incomplete oxidation of the glucose. Alright, and anaerobic respiration is also known as fermentation, means no oxygen is required. Alright, now fermentation is of 2 types, alcoholic fermentation and lactic acid fermentation. Whereas if the pyruvic acid enters the aerobic pathway, then complete oxidation of glucose takes place, and this takes place with the help of the Krebs cycle. Now first let's talk about the anaerobic respiration. Alright, so anaerobic respiration is of 2 types. First one is alcoholic fermentation and second one is lactic acid fermentation. So what happens during alcoholic fermentation? Pyruvate which is produced is converted into ethanol and carbon dioxide. Okay, alright, so ethanol and carbon dioxide are produced as byproducts. And what is the example of organism where alcoholic fermentation takes place? It takes place in yeast, okay, which is a unicellular eukaryote. And another thing to notice here is that there is a regeneration of NAD+. Okay, during glycolysis, NADH was produced. Okay, here NADH produced is utilized now and NAD is produced. Alright, so this was alcoholic fermentation. Net gain here is 2 ATP. Why? Because what is happening here, there is no ATP formation. ATP is not formed, only NAD plus is formed. Okay, this reduced coenzyme NADH is oxidized and no ATP is formed. But whereas 2 ATP, why we are writing 2 ATP? Because 2 ATP were formed during the glycolysis. Okay, this is the glycolysis steps of the glycolysis. Okay, and here 2 ATP were produced. Net output we have seen was 2 ATP. So net gain in fermentation and or alcoholic fermentation is 2 ATP. Now let's talk about the lactic acid fermentation. In lactic acid fermentation, what happens? Pyruvic acid is converted into lactic acid and again NAD plus is regenerated. Okay, regenerated. Alright, here again there is no ATP formation, so net gain is again 2 ATP. Alright, but the thing here to remember is that lactic acid, it provides a survival strategy during the intense exercise. Okay, during intense exercise, what happens? There is oxygen depleted condition is created. Okay, so examples in some animals you can say, like humans also, lactic acid fermentation can take place in the muscle cells. Alright, during intense exercise, and can also take place in some bacteria such as lactic acid bacteria. Alright, examples of lactic acid bacteria are Lactococcus and another can be Lactobacillus. Okay, now let's move to the aerobic respiration. Alright, so we have already talked about glycolysis. After glycolysis, what happens? Pyruvic acid, which is formed, okay, should enter the Krebs cycle, but pyruvic acid doesn't enter the Krebs cycle directly. What happens? Pyruvic acid is first decarboxylated. There is oxidative decarboxylation of pyruvic acid, which means that there is a release of the carbon dioxide. Okay, or you can say the carboxyl group is converted into the carbon dioxide group. Okay, and what happens? So pyruvic acid loses a carbon dioxide molecule and forms acetyl coenzyme A. So you can see here pyruvic acid, which was produced in the cytosol. Okay, it enters the matrix first. Alright, this is the matrix of the mitochondria. Alright, and here what happens? Coenzyme A is added. Okay, carbon dioxide is released and NAD is converted into NADH. Alright, so again this reaction is catalyzed by the complex pyruvate dehydrogenase complex and it requires magnesium ion. Alright, so what is the net gain of? We can say oxidative carboxylation. Net gain here again is 2 NADH. Alright, no ATP is produced here, but NADH is produced. There are 2 molecules of pyruvate. Okay, so 2 NADH will be produced. Okay, so net output or net gain is 2 NADH. Now the acetyl coenzyme which is produced can now enter the Krebs cycle. Now Krebs cycle is also known as the citric acid cycle because the first compound formed is a 6 carbon compound known as citric acid. Now Krebs cycle is also called TCA cycle, tricarboxylic acid cycle. Okay, it is also called tricarboxylic acid cycle. Why? Because there are 3 carboxylic group. Okay, 3 carboxylic group are present. That's why tricarboxylic acid. So what happens during Krebs cycle? First step is the condensation of the acetyl coenzyme A, which can be produced by glucose or by fatty acid depending on the requirement of the organism. The acetyl coenzyme A condenses with the 4-carbon compound oxaloacetate. Alright, and what happens? A coenzyme A is released and citrate, which is a 6-carbon compound, is synthesized. So this reaction is catalyzed by the enzyme citrate synthase. Now what's happened in the next step? Citrate is isomerized. So this is an isomerization step. Alright, a water molecule is released and isocitrate is formed. The enzyme involved is aconitase. Now isocitrate is further decarboxylated to produce alpha ketoglutarate, which is a 5-carbon compound. The thing here to note is that NAD is converted into NADH. There is oxidative decarboxylation, carbon dioxide is released. Enzyme involved is isocitrate dehydrogenase. Now next what happens? There is second decarboxylation and again carbon dioxide is released. Okay, NAD is converted into NADH. Okay, the enzyme involved is alpha ketoglutarate dehydrogenase, and it leads to the formation of 4-carbon compound, okay, known as succinyl coenzyme A. Now again what happens? Succinyl coenzyme A is converted into succinate, which is a 4-carbon compound, and here what happens? GTP is produced from GDP. So again this is a substrate level phosphorylation step, okay, and this GTP formation is coupled with the ATP formation. Okay, now again what happens? Succinate is further converted into fumarate and FAD is converted into FADH2. Enzyme involved is succinic dehydrogenase. Now fumarate is again converted into malate and malate is finally converted into oxaloacetate. So means there is regeneration of oxaloacetate and again NADH is produced. Alright, so this is the end of the Krebs cycle. But the thing here to note is that there is generation of NAD 3 times. First is here, second in this step, and third step is this one. Okay, and then what happens? One GTP is produced in this step and FADH2 is produced here. Alright, now this Krebs cycle occurs twice. Okay, it takes place 2 times. Why? Because 2 molecules of pyruvic acid was formed, so 2 molecules of acetate will be formed, okay, which can combine with 2 molecules of oxaloacetate. Alright. So now let's talk about the respiratory balance sheet. Alright. So what happens during aerobic respiration? First is glycolysis. During glycolysis what happens? 2 ATP were produced and 2 NADH were produced. Alright. During decarboxylation of pyruvic acid, again 2 NADH was produced. During TCA cycle, what happens? NADH was produced 3 times, FADH2 was produced 1 time, GTP was produced 1 times, and what happens? Krebs cycle occurs twice. Okay, so we have to multiply everything by 2. Alright, so this means 6 NADH, 2 FADH2 and 2 GTP. So now we know that 1 NADH corresponds to 3 ATP. Alright, 1 FADH2 corresponds to 2 ATP and 1 GTP corresponds to 1 ATP. So accordingly, what will happen? 8 ATP are produced here, 6 ATP are produced during oxidative decarboxylation of pyruvic acid, and 24 ATP are produced during the Krebs cycle. So what happens? In total, 38 ATP are produced during the Krebs cycle. But what happens? Sometimes 2 ATP are consumed, okay, during the transfer of NADH, which is produced during the glycolysis, okay, to matrix. Okay, so 2 ATP are consumed when NADH produced during the glycolysis. Okay, glycolysis takes place in cytoplasm. 2 ATP are consumed when NADH is transferred or transported from cytoplasm to the matrix. So what is should be the net output? 38 minus 2 will be 36 ATP. Alright. Now third one was the oxidative phosphorylation. Now what are the features or the components of oxidative phosphorylation? First one is electron transport chain, second is terminal oxidation, and then we have the chemiosmosis. Okay, now let's talk about oxidative phosphorylation. Okay, it is the process that harnesses the reduction of the oxygen, alright, to produce energy in the form of ATP. Okay, so here they are saying that there is a reduction of oxygen in the end, which is known as terminal oxidation. Okay, and this leads to the formation of ATP. So what happens during oxidative phosphorylation? Electron transport chain is taking place, okay, or the electron transfer is taking place. So what happens? The downhill transfer of electrons, okay, along the electron transport chain creates a proton gradient across the mitochondrial inner membrane. Okay, so what happens when the electrons are transported from one electron carrier to another electron carrier, from one to another electron carrier? Okay, and what happens? There is a transfer of electron from a molecule with low redox potential or reduction potential to a molecule with high redox potential or high reduction potential. So what happens? During this electron transfer, proton gradient is created, okay, across the mitochondrial inner membrane. And what happens? The breakdown of this proton gradient leads to the generation of ATP. Alright. And another thing here to remember, we will study this in detail in next slides. Another thing here to remember is that ATP synthase, which is known as the complex 5, okay, of the electron transport chain, acts as a channel through which protons flow back, okay, into the matrix of the mitochondria from the intermembrane space. And this, okay, transfer of the proton generates energy to generate, okay, or to synthesize ATP. Okay. So now let's talk about the electron transport chain first. So electron transport chain, okay, what does do? It consists of a series of protein complexes and electron carriers. Okay, there can be electron carriers or they can be hydrogen carriers also. Okay, and these carriers are embedded within the inner mitochondrial membrane. Okay, and these carriers and protein complexes, what they do? They facilitate the downhill transfer of the electrons. And what happens during the downhill transfer of electrons? Okay, during the sequential transfer of electrons, a proton gradient is generated that drives the synthesis of ATP through complex 5. Now there are two kinds of electron carriers, or okay, two types of carriers. First one is hydrogen carrier, second one is electron carriers. Okay, FAT is an example of hydrogen carriers. Cytochrome B and cytochrome A and A3 are the example of electron carriers. And ubiquinone acts as a hydrogen as well as the electron carrier. Okay, so now let's talk about electron transport system in detail. So electron transport system consists of 4 complexes. Okay, we have complex 1, complex which is known as NADH dehydrogenase. Second complex is known as succinate dehydrogenase. The third complex is known as cytochrome BC1. And the complex 4 is known as cytochrome C oxidase. Okay, here in this figure, you can see the electron transport system. Here we have the complex 1, then we have the complex 3, then we have complex 2, then we have complex 4, and later we have the complex 5, which is known as ATP synthase. Okay, this is also known as complex 5, and this complex is involved in ATP synthesis. Alright, and where does this electron carriers are present? They are present in the inner mitochondrial membrane. So this is inner mitochondrial membrane. Alright, now let's talk about the complex 1, which is known as the NADH dehydrogenase. So what happens? This complex, okay, transfers electron from NADH, okay, NADH, which is a reduced coenzyme, okay, and where were these NADH produced? They were produced during glycolysis and Krebs cycle, and also during oxidative decarboxylation. So now what is happening? The reduced coenzymes are being oxidized, and the electrons from the NADH is transferred to ubiquinone. So this is ubiquinone. Okay, and ubiquinone finally transfer electrons to ubiquinol. Ubiquinol is nothing but reduced form of ubiquinone. Now you can see NADH dehydrogenase. Okay, what is it? Is an FMN, protein, flavin mononucleotide, and it is associated with the iron sulfur protein. So here you can see electrons are transferred to the ubiquinone and finally to ubiquinol. Alright, this we have already discussed. Okay. Now complex 2, which is known as succinate dehydrogenase. This complex, what it does, transfer electrons from succinate. Okay, from succinate to ubiquinone and then again to ubiquinol. Alright. So this enzyme, consists of flavin adenine dinucleotide, and it is associated with the iron sulfur protein also. It transfers electrons from succinate to ubiquinone and then finally to ubiquinone and then finally to ubiquinol. Now let's talk about the complex 3, which is known as cytochrome BC1. So what does this do? This receives electron from the ubiquinol and transfer it to cytochrome C. Okay, so what does this complex do? This receives electron from ubiquinol. This is ubiquinol. Okay. Alright, and what does it do? It transfers electrons to cytochrome B. This is cytochrome B. And finally, electrons are transferred to cytochrome C1. Alright, so complex 3 is cytochrome BC1. Why? Because from ubiquinol, electrons are transferred to cytochrome B. From cytochrome B, electrons are transferred to cytochrome C1. Okay, and from cytochrome C1, electrons are transferred to cytochrome C. And what is cytochrome C? Cytochrome C is a mobile element. It can move. And what does it do? It connects complex 3 with complex 4. Alright, here we have cytochrome C. What does it do? It takes the electron from cytochrome 3, from cytochrome 1, and it transfers electrons to complex 4. Now this complex 4 is known as cytochrome C oxidase, and it consists of two cytochromes, cytochrome A and cytochrome A3, and there are two copper centers. One is this one and another is this one. Alright, and what is a unique feature here? The electrons here are transferred from cytochrome A3 finally to molecular oxygen and water is produced. So this is known as terminal oxidation. The oxygen, what does it do? It accepts protons and electrons as well as protons and electrons, and it gets converted into water. Alright, so complex 4 consists of two copper centers, and what it also consists of two cytochromes, cytochrome A and cytochrome A3. Alright, again we have discussed about this, and what is happening during the downhill transfer of electrons. Now electrons were moving from one complex to another, and what is happening along with the transfer of electron? Protons are moved across the membrane. So if this was the inner mitochondrial membrane, alright, this was the matrix, this is the intermembrane space. Alright, this was the intermembrane space. So protons were accumulated here initially. And what happens during the transfer or downhill movement of electron? Electrons are transported from the matrix to the here intermembrane space. So protons accumulate here. Alright, so if we look at this figure, so what is happening during the downward transfer of electrons? Protons gets accumulated in the intermembrane space. This is intermembrane space. This is matrix of the mitochondria. This is intermembrane space. Okay, now what happens? This was complex 4. Okay, oxygen accepts the electrons and protons, get converted into water, and what happens? Here, complex 5 we were talking about, which is known as the ATP synthase. Okay, it consists of two components, F0 and F1. So this F0 component acts as a transmembrane channel through which protons ion, you can see proton ion, okay, they move back to the matrix through F0. And what happens when they move back to the matrix? Okay, then there is a conformation change in the F1, and this conformation chain leads to the or generates energy for the synthesis of ATP. Alright, so this is known as chemiosmosis. Okay, formation of ATP, okay, by the breakdown of the proton gradient is known as chemiosmosis, and this hypothesis is known as chemiosmotic hypothesis given by Peter Michel. Alright, so okay, let's talk about the complex 5. Complex 5, okay, consists of F0 and F1. Alright, here you can see there is a breakdown of proton gradient. Proton move back to the matrix, okay, through F0, which is known as the transmembrane. It is a transmembrane ion channel. Okay. Alright, it is a transmembrane ion channel through which protons move back to the matrix, okay, and leads to the generation of ATP. And what happens? There is a conformation change here. There is a conformation change in the F0, which leads to the synthesis of ATP from ADP and inorganic phosphate. Alright. F1 is also known as the headpiece. Okay, it consists of the catalytic site. There is a conformation change or the mechanical rotation in the F1 component of ATP synthase, okay, as the proton pass, which leads to the formation of ATP. Now, terminal oxidation, we have already discussed terminal oxidation. What happens? Oxygen, okay, it accepts electrons and protons, okay, and there is a formation of the water molecule. And why it is known as terminal oxidation? Because it is the final step, okay, in the process of electron transport chain. All right. Now let's talk about the amphibolic pathway. All right, amphibolic pathway, again, respiration we usually think is a catabolic process, but actually respiration is a amphibolic pathway, means it involves both anabolism and catabolism. All right, and depending on the situation of the organism or the requirement of the organism, okay, either anabolic pathway can occur or catabolic pathway can occur. All right, so what happens? Okay, we know that during respiration acetyl coenzyme A is produced, okay, due to oxidative decarboxylation of pyruvate, and pyruvate is produced, okay, as the end product of glycolysis. This pyruvate can either be produced, okay, it can be produced by glucose or fatty acid. Alright, pyruvate is converted to acetyl coenzyme, then acetyl coenzyme enters the Krebs cycle. Alright, so if acetyl coenzyme A enters the Krebs cycle, it will be a catabolic pathway. Because what is happening? There is a breakdown of complex material into simpler one. Okay, that's why catabolic pathway. But if the organism has ample amount of energy, then what is happen? Reverse can happen. Okay, acetyl coenzyme A can again be converted back into the fatty acids. Okay, and fatty acid will later form lipids. All right. So either catabolic mechanism can activate or anabolic mechanism can activate depending on the requirement of the organism. All right. Now another thing is that respiration is interconnected with various metabolic pathway. Okay, it is not a solo pathway, but it is connected to various metabolic pathway such as lipid synthesis, okay, protein synthesis, all right, and formation of other organic molecules, because the intermediates of the Krebs cycle can lead to the formation of other organic molecules as well. All right, because the respiration is interconnected with other various metabolic pathway, this can lead to either catabolism or anabolism depending on the requirement of the organism. Now let's talk about the respiratory quotient. So respiratory quotient, what it is? It is the ratio of the carbon dioxide released to the volume of oxygen consumed. Okay, carbon dioxide which is released, okay, and the oxygen which is consumed. All right, oxygen consumed. Okay, so the value of respiratory quotient depends on the substrate, okay, or the nature of the substrate. Okay, now the RQ value for carbohydrate, if it is undergoing aerobic respiration, then what happens? Okay, if this is the glucose molecule, all right, oxygen is required for the breakdown of the molecule, and 6 carbon dioxide are produced, water is produced, as well as energy is produced. So RQ value here will be 6 divided by 6 means 6 carbon dioxide divided by oxygen consumed, okay, which is 1. Okay, so it is unity, okay, for carbohydrates which are undergoing aerobic respiration. Now RQ value for carbohydrates undergoing anaerobic respiration. In anaerobic respiration, what happens? No oxygen is, okay, used up. So glucose, okay, there is no oxygen, produces alcohol, ethanol, and carbon dioxide produced as a byproduct. Okay, then what happens? RQ value here will be infinity. Why? Because 2 carbon dioxide divided by, there is no oxygen consumed, okay, which gives the value of infinity. Now RQ value for proteins is around 0.9 and RQ value for fats is around 0.7. Okay, so RQ value for proteins and fat is always less than unity. Why? Because both proteins and fat require more amount of oxygen, okay, to burn, okay, and the fats, they require even more oxygen than proteins. That's why RQ value is less, okay, it's 0.7 as compared to 0.9. All right, so this is an example of, okay, RQ value of fat. This is tripalmitic acid, okay. See, the amount of oxygen used is very high, 145, okay, carbon dioxide produced is this much. All right, so RQ value will be 102 divided by 145, which is around 0.7. Another thing you have to just remember that RQ value for organic acids, okay, you have to remember is greater than unity. Okay, this you have to remember. So this was the end of this session. Hope you enjoyed it and thank you for listening.